Cellular basis of life. Cell, as we said before, is the basic unit of life. Every living organism has at least one cell. Many have many, many cells. Everything smaller than a cell that's not a cell is not living. So, a virus that's sitting on the desk is not alive. A virus that enters a cell and hijacks it and uses it as its own cell becomes living, right? A cell that's infected by a virus and controlled by a virus is now a living organism. When a virus goes out and sits on a desk again, it's not living anymore. So everything else has cells. What is a cell? Well, cells come in all shapes and sizes. The largest cell ever is the egg of an ostrich. Of an ostrich is a pretty big thing, and it's apparently the biggest egg ever. It's bigger than the eggs of the biggest dinosaurs. Maybe there were some extinct birds that were bigger and had bigger eggs, like Moa and Epiornis, but we don't know that. Some of them are very, very long. For instance, the sciatic nerve in the hind end of the giraffe might be as long as 3 meters. Uh, Nerves in some other animals may be even longer. For instance, the dorsal root ganglion in the blue whale may have the longest axon going from the brain all the way to the tail. So that will be the longest uh, cell today. Uh, when we look at dinosaurs, like the enormous sauropod dinosaurs, they may have uh, had even longer nerves. For instance, the recurrent laryngeal nerve, which kind of winds its way through the body, going back and forth. Could have been 92 feet in Supersaurus, or up to maybe even 190 feet in length in Amphicelius. But those are dead now. So currently, the longest possible cell in a currently living organism is the dorsal root ganglion in the blue whale. But it's very, very thin. An ostrich egg is much bigger and heavier than that skinny little nerve. What is a cell? A cell is a small packet. It's a bag of liquid. Inside of a cell is cytoplasm or cytosol, which is essentially salty water and various organic molecules that are suspended in it. Another way to put it is the cell is a bag of gunk and goo, which is just floating around. Uh, so the cytoplasm is contained, that liquid is contained within a cell membrane. Cell membrane is a phospholipid bilayer, which means that it is composed of two layers of tightly packed molecules of fat. So there are proteins embedded in that fat layer, and those proteins can kind of freely float around that membrane back and forth, move around, bind to each other, unbind. And they're very important for the communication that has to happen between the inside of the cell and outside of the cell, all the signaling between one cell and the next, or between the cell and the environment. So these proteins embedded in the membrane are important. On the outside of the membrane, some cells have various other structures. We don't, as animals, we don't, but some bacteria and some plant cells have thick cell walls. They're kind of thick organic layers on the outside of the cell membrane. They give that membrane some strength and rigidity, so the cell is better equipped to defend itself against various, mechanical or chemical or biological attacks. Some cells have cilia on the surface. Those are kind of hair-like structures, uh, or pili, which are a little longer, or a single long whip-like flagellum at one end. So, for instance, a sperm cell has a flagellum with a long tail at one end of the cell. Cilia we find in number of parts in our body, including, for instance, in trachea and bronchi. So those structures allow the cell either to move using its own energy, or to move other stuff across its surface. So, for instance, a sperm cell is going to use the flagellum to move itself, while the cells in the trachea are going to use the cilia to move stuff along its its surface, moving all sorts of dust and pollen and bacteria that come into the respiratory system and push it out into the throat. Inside of every cell, there is hereditary material, which is DNA. There are some exceptions to that. For instance, red blood cells in our bodies and on most mammals uh, have a membrane in cytoplasm, and they have packed molecules of hemoglobin, but there is no nucleus. Nucleus is kicked out of that cell during development, so there's no hereditary material in red blood cells. They can't divide; they have to be made anew. There are exceptions to this. For instance, camels have red blood cells with the nucleus. The nuclear material, the DNA, is inside of the nucleus and is organized in a form of multiple chromosomes. Uh, we're going to look at them a little bit later, but you also covered all that in basic biology courses or if you had genetics courses. Eukaryotic cells, unlike bacteria and archaea that are prokaryotes that have no organelles, eukaryotic cells have organelles. An organelle is a subcellular structure. And what it means is that it is usually a membrane or a set of membranes that provides a separate compartment inside of the cell. So most organelles are made of membrane that is very similar to the cell membrane. It can be either a single or a double layer of phospholipids, and then there's going to be some pores and some proteins that are going to allow particular molecules to go in and go out of those compartments. This allows the cell to store particular molecules in one place, to prevent those molecules to be degraded by enzymes elsewhere in the cell, all sorts of reasons to separate some biochemical reactions from each other so they don't interfere with each other. So we have compartments, and each compartment you can have different things going on, and they are free to go on without interfering with each other. Nucleus is the largest membrane-bound organelle. Most cells in our body have only a single nucleus. Some have multiple nuclei. For instance, our heart muscle cells have multiple nuclei. Um, the function of a nucleus is to remove the DNA from the rest of the cell. So you don't have, all sorts of things happening right there on the DNA, and maybe in some way, damaging the DNA. The nuclear membrane or the nuclear envelope is also phospholipid bilayer, and it selectively allows molecules to pass in or out the nucleus, between the nucleus and the cytoplasm. So, for instance, messenger RNAs can go out of the nucleus but can't go back in. On the other hand, small molecules like, for instance, steroid hormone receptors can go from the cytoplasm into the nucleus. They're allowed there. There are pores that control what can go in and what can go out of the nucleus. Inside the nucleus, DNA is organized chromosomes. Chromosomes are tightly coiled and wound strands of DNA packaged with various proteins like histones. Endoplasmatic reticulum, there are two types. There is rough and smooth. Rough endoplasmatic reticulum, it's a bunch of membranes kind of in parallel with each other, on top of each other, like Philadelphia. If you have Spanakopita, it looks like that. And inside of those membranes are embedded ribosomes, which is an organelle, a structure made of proteins that are embedded in that membrane of the rough endoplasmatic reticulum. It's called rough because under the microscope, ribosomes make it look like rough. And ribosomes are structures that read the code from the RNA and use that to put together amino acids into this into a chain, thus starting the process of the production of proteins. Smooth endoplasmatic reticulum is the same kind of thing, except it doesn't have ribosomes. Between those layers, between those phyllodo membranes, are spaces that are mostly uh, protected from all sorts of other chemicals coming in. And this is where we have other things occurring, for instance, the synthesis of lipids and synthesis of carbohydrates. Once those things are made, various types of proteins and lipids and carbohydrates, they often need to be stored somewhere. Golgi apparatus is excellent for storage. It stores and packages nicely, tightly, and neatly various molecules. So when a molecule is needed elsewhere in the cell, a portion of that membrane uh, compartment of the Golgi closes off and forms a vesicle, and that vesicle can then be then transported to a different part of the cell where it opens up, releases those molecules, and those molecules can then be used by that other part of the cell for whatever it needs to do. There are some organelles that contain a little bit of their own DNA. So those are chloroplasts in plants and mitochondria in animals. So those are so-called endosymbionts. They started as cellular parasites billions of years ago. A bacterium, a smaller bacterium, entering inside of a bigger bacterium and being just like a internal parasite using all the materials there. But that's an internal parasite that had some of its own uh, wonderful functions that it could do, and so over billions of years, those two cells, the parasite cell and the host cell, evolve, which often happens, evolve from parasitism into some symbiosis. During that process, most of the genome of the parasite cell got transferred into the nucleus and the genome of the host cell. But a few genes remain, so there are a few genes in mitochondria which are expressed inside of mitochondria, then taken, of course, to the rough and the plasmatic reticulum to form proteins, but it's a different kind of regulation because they're not in the nucleus, they're in the mitochondria, and they're needed for the proper function of mitochondria. So there's some DNA that's not in the nucleus, not much of it. We can use this, for instance, because mitochondria are mostly 99.99% of the time uh, transferred only from the mother to the offspring. So we can use the sequencing of the mitochondrial genes to infer uh, origin through the maternal line. Uh, Mitochondria are energy factories of the body. Uh, what is happening there? So, when we eat different kinds of foods, we are going to eat a bunch of sugars. Our digestive system, including the liver, are going to break all sorts of other types of sugars and convert them into glucose. That glucose is going to be stored as glycogen in various places, and then when it's needed, the glycogen is broken down. Put the glucose molecules are put in the bloodstream, and that glucose goes into all the cells where they're needed. For instance, in actively working muscles or wherever they're needed. So the cell gets those glucose molecules and starts breaking them down. The first part of the breakdown of glucose molecules is glycolysis, which is happening in the cytoplasm. It has only a couple of metabolic steps. It requires two ATP molecules to get this whole process started, but then produces four ATP molecules as a result. So we get a net gain of two ATP molecules. This is anaerobic respiration. This is doesn't require oxygen. It's easy. It's quick. It results in the production of lactic acid, which can then be transferred by circulation back to the liver, where the liver can break it down and make more glucose out of it. Uh, but the breakdown product of of glycolysis then moves into mitochondria. And inside of mitochondria, it undergoes a whole kind of sequence of biochemical reactions that you have probably heard of as Krebs cycle or citric acid cycle. And so glycolysis is the first process, the first step. Krebs cycle is the second step, and the last third step is the electrons transfer, uh, which finishes the breakdown of glucose. At the end of that process, when the glucose molecule has been broken down, what we have is all those carbons and oxygens and hydrogens from the glucose are somewhere else. They are in molecules of water. No big deal. Cells are made of water, right? If it's too much water, it can leak out of the cell into the circulation, to kidney and out, or it can be taken to sweat cells and out. So making a little bit of extra water is no big deal. It also produces carbon dioxide. In order for this to happen, we have to have oxygen inside of those mitochondria. This whole breakdown of glucose cannot happen without oxygen. That is why we have respiration. That's why we have breathing. We have to inhale oxygen, which then goes into all the cells, into the mitochondria, and it's essential for the proper breakdown of glucose molecules. As a result of that, this process generates carbon dioxide. We don't need carbon dioxide in our cells, so it goes into the circulation, gets taken to the lungs, and we exhale carbon dioxide. What else is produced? Energy, lots of energy. And that energy gets bound into those three bonds of the three phosphate groups of adenosine triphosphate, or ATP. So, if you look at the textbook, it says that breakdown of one molecule of glucose is results in the production of 36 molecules of ATP. Well, some say 38. However, that energy has to also get used for some other processes as we go, replenishing some of the enzymes and whatnot. So, really, a breakdown of a one molecule of glucose ends up generating about 30 to 32 molecules of ATP. That's real. Theoretically, it should be 36 to 38. Uh, in reality, it's about 30 to 32. So, if I ask you the question, how many molecules of ATP are generated by breakdown of a single glucose molecule, any number between 30 and 38 I'll take as correct, because they all can potentially be correct. Well, if you think of mitochondrion as an engine, remember it's an energy factory of the body. Everything, every engine that you can think of is not 100% efficient. You look at your engine of your car after you ran it for a few minutes and it's hot, so some of the energy and the chemical bonds of gasoline, instead of being used for turning wheels, gets released as heat instead. So it's a loss of energy, and every engine is inefficient. There's no such thing as 100% efficiency. Then you go down to the molecular level and biochemical level. There are all sorts of other things that are going to make it less efficient. There's going to be, a random bumping of molecules into each other, which means that the breakdown of glucose by enzymes during glycolysis and Krebs cycle is not going to be perfect. It's not, sometimes it's going to work out just right and sometimes it's not. Just because a chemistry book says that this enzyme cleaves this off of that molecule and then adds phosphate to this molecule doesn't mean it always happens that way. It has to be everything aligned just perfectly, and there's going to be some noise in that system. There's going to be some inefficiency. Not every molecule of glucose is going to be broken down properly, which means not every molecule of glucose is going to produce those 30 to 38 ATPs. Uh, and as a result, they're not going to produce carbon dioxide and water. They're going to produce things like hydrogen peroxide instead. Hydrogen peroxide is a free radical. Free radicals are extremely reactive small molecules, and they are damaging the cell, and that's part of an aging process. So these reactions that produce free radicals are damaging the cell because free radicals are going to react with whatever they first encounter, which can be a protein, an enzyme, a membrane, whatever. Um, so that is intercellular damage by free radicals caused by an inefficiency of the mitochondrial engine, which means not every glucose molecule is perfectly broken down. So sometimes the breakdown products are not, carbon dioxide and water we know how to deal with. There are other molecules which are dangerous. So some animals, those are birds and mammals, warm-blooded animals, they've evolved a way um, to manage this process. That some of the energy that is released from the breakdown of glucose, but the imperfect, erroneous breakdown of glucose, instead of going into the ATP, is released as heat, right? There's no other way. So you're breaking down all those chemical bonds of glucose, and instead of making ATP because there was some error, that energy gets released as heat. Actually, the inside of mitochondria is warmer than the outside of it in the cytoplasm. And so birds and mammals have evolved mechanisms to control this, to control the efficiency of the engine that is mitochondria. How does it do that? It opens and closes pores in the membrane of the mitochondria. If the pores are open, then the whole process of breakdown of glucose is less efficient, thus producing a little bit less ATP but more heat. If you close the pores, there is more ATP made but less heat. What opens and closes those pores? Well, thyroid hormones open those pores. So thyroid hormone is going to decrease the efficiency of the breakdown of glucose inside of mitochondria, resulting in higher release of heat. That is how birds and mammals control the body temperature. They control the heat. So if you have an unnaturally low body temperature, you go to the doctor. The first test that the doctor orders is a thyroid test because something's wrong with your thyroid gland or the regulation of the thyroid gland, and it's not secreting thyroid, and thus your mitochondria are not opening up the pores. So everything is going towards work building ATP, and very little is released as heat. So you are not warm enough. So this is something to keep in mind when you have thyroid problems, that it is going to decrease body temperature. We call that, decrease in metabolism, but decrease in, it's really just a decrease in body temperature. Finally, the cell have other structure, which are called, under one umbrella of the word cytoskeleton. So those are different kinds of filaments and microtubules. They anchor the organelles in a particular place. They give cells shape, kind of like a skeleton of a cell. They can move molecules and organelles and vesicles within the cell from one place to another. So there's a lot of complicated stuff about it. You don't need to know a lot of detail in this course about the cytoskeleton. So this is where I'm going to stop about the cells, and we're going to continue with molecular stuff in the next video.